All right. So as Arthur mentioned, I'm Michelle Montgomery. I have been working with little fire ants for over a decade, and this was it was a great opportunity offered to me to be able to conduct my PhD research at the University of Canberra. And I did my thesis on developing management strategies for little fire ants in the tropics. So I've focused on using Hawaii as my study site because that's where I live, and obviously it's very accessible here. And we have lots of ants. I first want to say thank you to everybody that has helped me along the way, all of my advisors, especially the different backgrounds that all my advisors have. It has brought a lot of feedback and I feel like I'm a much more well-rounded researcher now with your guys' guidance. Everyone from the Hawaii Ant Lab, past and present, who has helped along this way and for the various projects, and also our collaborators on a lot of these projects. So jumping in, my thesis structure is, hold on one second, I need to. Okay, so my thesis is comprised of 5 peer-reviewed publications, which 2 of which are currently in review. Today, I'm really going to be focusing mostly on the applied research aspects of the thesis. I could talk about ants all day, every day, they do, but for the sake of time, today you guys are going to get a really abridged summary of the research that I've done within this, mostly within the thesis, but also a little bit outside of the thesis that's relevant. I've tried to make sure that there's plenty of time for discussion afterwards, but I do have a meeting right after this. And so if for whatever reason there isn't enough time, I will make sure to put my email into the chat, and you guys are always welcome to reach out to me anytime if you want more detail or just to talk story about any of this stuff about adaptive management in general or about little fire ants specifically. So my research for my thesis fits into this larger applied research framework. The framework includes academic and applied research, and it can be broken down into as many parts and subparts as you like. This is just an example of the framework that I'm working within, which includes both lab and field experimentation for the applied research aspect. I've done quite a bit more work within this framework that's not actually included in the thesis, and I'll briefly touch on some of that as well, so you kind of see where all of this is going and you're not just getting these little snippets here and there. And by following this kind of a framework, you can guide your research into a logical progression towards an ultimate goal of developing species-specific management plan. And so first I'm gonna have to set the stage for you a little bit, so you know why all of this is important. I'm not gonna go into too detail of the background because these guys get crazy. I think everyone here knows that invasive species are a big problem. They're really bad. One reason that is that they're very difficult to control once they're established, and they quickly spread. Some taxa, such as ants, are more difficult to control than others, and this is for a variety of reasons. Some of these species may not have effective control options available. Maybe they're new and emergent invasive species that haven't really been known to be invasive in the past, or maybe it's because there's established invasive species that generally are overlooked by national and local governments, and so there's not a lot of focus being put onto them. Because of these specific things, my research is really centered on species-specific management, and this kind of research is essential for the future of controlling invasive species. So my focus is on invasive ant management specifically, not just general invasive species. Invasive ants are considered some of the most devastating insects that are, and they're notoriously easy to transport. They're very difficult to detect, and as social insects, they're much more likely to quickly establish and thrive compared to other insect taxa as they are introduced into new locations. The term tramp ants that you see here, this is really given to a specific group of species that they're commonly transported around through human activity. They're very closely related with human activity. And they also share these uncommon biological and behavioral traits that aren't really seen very much in the vast majority of ant species. It's these traits that are often, that's believed to really further increase the chances of successful establishment, of spread, and the magnitude of the negative impacts that they impart. The target species that I work on is little fire ants, and it's also called electric ants in Australia. There are a dozen other names, common names for them, depending on where you're at in the world. I'm going to be referring to them as little fire ants. This species is native to Central and South America, but it is widespread throughout the tropics, mainly in the Pacific region. Now, however, this is a highly adaptable ant, and it will also invade subtropical and Mediterranean climates when it is introduced, and it is also a known greenhouse pest of temperate climates. So as climate zones are believed to shift in the wake of climate change, it's very possible that we may find the potential range expansion for little fire ants is going to leave very few safe places on the planet when it comes to little fire ant invasion. There may not be a whole lot of areas that are, quote, immune to this little bugger. It's a very tiny species. It's only about 1.5 millimeters. You can see over, I'm not sure if you can see my cursor, but over here on the tip of a pencil, that's how small these guys are. They build very small individual nests, as you can see here. This is just a portion of a macadamia nut shell. This can host eggs, larvae, workers, queens, all in a full nest, all in this tiny shell. But they also possess, the typical features of a tramp ant, which leads them to be able to build these super colonies where ants from individual nests work together and they share their resources. This is because they have multiple queens, they're unicolonial. So this in particular is where they build their super colonies. All of these workers go in and out of other nests and they share their resources for success. Super colonies can support extremely high population densities within an area, and they're easily fragmented by people moving items in and out of infested areas. Their small size and their rusty color makes them very difficult to spot, even when there's a ton of ants present on items being moved. In addition to the typical tramp ant features, such as polygamy, unicoloniality, they also reproduce clonally. This clonal reproduction even further increases the chance of successful establishment, even when there's a few ants that are moved around, because the queens don't necessarily have to mate in order to be able to produce viable offspring. The little fire ants impart multi-tiered negative impacts, both directly and indirectly on people and the environment which they invade. They sting people and pets. It's common for animals to go blind within the areas, infested areas. They farm agriculture pests, which can vector plant disease. There's anecdotal evidence that they actually impact vertebrates. This picture of the gecko, this gecko jumped into my laboratory colonies and got swarmed. So this is actually something from our lab. It also really impacts, there's pretty hefty economic impacts from the species. It does impact tourism, property values. You have to declare that this pest is on your property if you're going to sell it. The cost of treatment can be very high, and it really downgrades the quality of life when you're living in an infested area. As with other tramp ants, the little fire ants are closely associated with humans. They are moved around by people. The common pathways in Hawaii specifically, and likely elsewhere, are through the movement of, say, nursery stock, the friendly sharing of propagative materials. Everyone here likes to share their coconuts, and that's a big way that these guys get moved around. They move around in vehicles. Vehicles will be infested and sold, or just driving one place to another. Shipping pallets are frequently infested, and even household items. So couches, furniture, stuff like that. And when people pack up their house and move to a new location, they don't really think about disinfesting their belongings before they move. And so they can bring these ants to new locations. Globally, the spread of little fire ants is strongly associated with international trade. We know this because the species reproduces clonally. We can trace the genetic lineages of the clonal populations, and that's what you see here in these pretty little colorful arrows, where the same genetic lineage has been tracked. And these are areas and countries that have very close trade ties. In Hawaii, it was first detected in '99. It was suspected of being here for at least 5 years before, and early attempts to eradication were unsuccessful. And so it's basically had about 30 years in order to spread throughout the islands. Historically, when it comes to ant control, insecticidal baits are widely considered the most effective option. Commercial, quote, fire ant baits are generally granular. They're made up of a corn grit soaked in soy oil with a tiny bit of active ingredient. Most commercial fire ant baits are very attractive to little fire ants, and they've been assumed to be effective. They were used during the original eradication attempt in the Galapagos and during the early eradication efforts in Hawaii. Unfortunately, we don't have any real update on the status of the Marchena Island eradication. So I don't know if there's any follow-up data on that, but for Hawaii, it was clearly unsuccessful because it spread for the past 30 years. So the assumptions made by the early practitioners regarding the species in Hawaii, their assumptions on product stability, the treatment strategies and stuff like that were the main reason why early eradication attempts in Hawaii failed. when it comes to ant management, there's this sweet spot that we're really trying to hit. And if there's knowledge and understanding in any one or more of these vital areas, if that knowledge or understanding is lacking, we will miss that sweet spot and the chances for failure will increase. And so my job in the Hawaii Ant Lab and in this thesis is really to hit that sweet spot in order to find solutions to practical problems, in this case, little fire ants, through applied research. And to do this, I work within this, this framework that I mentioned earlier. The work that I present in my thesis really builds off of past works and my work will then be built upon in the future as research within this framework and on this topic continues. So in 2009, my boss, Cass Vanderwood, came up with the idea of this homemade gel bait that could be applied as a broadcast on the ground and in the trees. The bait was highly attractive to little fire ants and it filled these niches that granular baits did not. The application to vegetation, the sensitivity to moisture, these are the kind of niches that this bait matrix fulfilled. We call this the HAL gel bait for Hawaii Ant Lab. Much of the work that I do is really geared towards improving the formulation and finding and registering active ingredients for use in this bait, for testing application methods for the bait, and figuring out how this and other baits are best used when treating different types of infested sites. Laboratory and field experimentation are essential components of this overall process. And so in my thesis, while working within the framework, the experimental lab component of my thesis is really focused on these two things, really looking at how laboratory methods influence laboratory methodology in general, including raising little fire ants, really influence experimental results, and also trying to find a suitable insect marker so that we can do more resource allocation studies and stuff like that. First, I'm going to talk to you about about something that I thought may be confounding results in our lab experiments. This was a big issue. So as I do laboratory experiments, I need to do laboratory experiments in order to, screen insecticides, make sure that the bait is palatable, is effective before putting it out in the field. It allows us to screen a lot of different things in a fairly quick manner. But it's also well known that laboratory raised insects tend to behave differently from their wild counterparts. Because of this, the results from lab experiments shouldn't necessarily be viewed as a definitive representation of what to expect in the field. My experience in the past with raising little fire ants in the lab confirmed this, and I had noticed that the ants did not respond to the lipid-based fire ant baits, including the HAL gel bait, like the wild ants did. I wondered if maybe the rearing diet could be influencing this. This is a big deal. If you can't get realistic behaviors, you can't elicit a realistic behaviors from the ants when you're testing potential treatment options, then you're not going to be able to keep moving forward in a progressive manner during the research process. To test the idea that is the rearing diet responsible for these differences in behaviors, I conducted a series of dietary experiments in the lab where colonies were fed nutritionally defined diet of either high carbohydrate, high protein, high lipid, or giving them a balanced buffet style of diet. And after they were on this diet for a while, I tested their response to carb, protein, and lipid foods. I also conducted a food preference trial using the same carb, protein, and lipid foods in the field so that I could compare laboratory versus wild ant responses. Both of these, the laboratory and the field component, were comprised of a multi-choice and a no-choice experiment. So the multi-choice experiments allow us to see what the ants prefer when they're given a choice of different foodstuffs, while the no-choice experiments allow us to see that if the ants will respond well to a particular bait, say in this case the HAL gel bait, if no other option is available, and even if it's not their preferred food. So even if they have an option, if they don't really like it that much, well, if they don't have an option, are they still going to go for it or not so much? That's what we wanted to see here. The results during the lab experiment were really unexpected. For the most part, the preferences really shifted towards carbohydrates and away from lipids, regardless of what the dietary treatment was. So in this chart here, in this graphic, this is all change in recruitment. So we did a pre-treatment measurement, we did a post-treatment measurement, and we wanted to see what the magnitude of change in recruitment to the same things were. And this is what we got. The most interesting thing that I see here is the colonies that are raised on the sugar-based diet had the greatest increase in response to sugar, carbohydrate-based foods. And that was pretty opposite of what we thought would happen. So when we wanted to see, okay, well, what about if we didn't give them an option, if we didn't really care what their most preferred food was, are they still going to respond to the HAL gel bait? Because this is becoming a very widely used and preferred bait. What we saw was even though the response fluctuated over time, the general trend over time showed that the colonies maintained without lipids in their diet responded better to the HAL gel bait than colonies where their dietary treatments all included some kind of a lipid. What's more was that colonies that were provided lipid into their general diet showed a dramatic decrease in their recruitment to the HAL gel bait over time. So what this shows us is that if we were limiting the lipids in the gel bait, we would get a better response to, or if we limited the lipids in their diet, we would get a better response to the gel bait. And so if we wanted, we can compare that to what we see in the field where wild ants above and beyond prefer the gel bait, the lipid-based bait over anything else, regardless if they have a choice or not. They really like those oils. So this was a pretty drastic difference in behavior that that we needed to document and try to rectify. So ultimately, in the end, at the end of the day, we still really don't know what exactly is responsible for the behavioral differences, but we are able to elicit, some kind of a reasonable response from the lab ants by omitting lipids from their diet for a couple of weeks and providing a sufficient fasting time right immediately before conducting any kind of feeding experiment or the measurements during a feeding experiment. Okay, moving on to the second part of the laboratory experiments that I conducted. This one was looking at potential markers for little fire ants. Now, insect marking is a valuable tool when researching a variety of topics. There are a lot of different kinds of markers available, but not all markers are suitable for certain taxa, especially minute ants. At the moment, I'm really focused I'm interested in foraging, how food and baits are shared throughout the colony, or how it's shared throughout the landscape, potential non-target impacts from bait treatments. And since I'm working with tiny ants that share and dilute resources, I chose to look at the animal proteins as markers, since they're fairly cheap and they're easy to identify or detect in very minute concentrations through ELISA analysis, which is enzyme-linked immunosorbent assays, in case you didn't know. So really, this is the first work that's been done on any kind of marking techniques for little fire ants. So this work is really like proof of concept and identification of possible problems. For the thesis, I looked at the use of animal proteins as a topical marker. And the good news is that it worked. The bad news is some of the markers were not retained for very long, so 24 hours tops, and cross-contamination was actually really high. Now, the cross-contamination issue wasn't overly surprising because it is an external marker and ants are physically interacting with each other all of the time, including they groom each other. So if you spray them with a marker, if they have a topical marker, there's a high chance that they're going to pass it to each other. Not included in the thesis is some additional work that I've been doing looking at these animal proteins as internal markers. This has been a really interesting tangent that's fascinating to me, and it's really helped us understand not only the marking, but also the actual bait matrix that we're working with. So I've been running into some challenges during this process, namely solubility of the marker. So the Hal gel bait is a lipid-based bait, and most active ingredients that you would use in an insecticidal bait are water or are also lipid soluble. In contrast, the markers that we're using are water soluble, and so they're not necessarily well transferred when you use them in our gel bait. Because the gel bait is a homemade bait, there isn't these formal and fancy emulsifying agents. So if there's any kind of the tiniest bit of separation between thickening agents and the oil, the vegetable oil that's in there, that the ants will be feeding on that and not taking up the marker. And so that's been challenging. I'm currently working on finding solutions to that problem, and I'm in the final throes of that. And then we'll be shifting over to doing some field trials, looking at how, for example, looking at how if you set out a bait station, how big of an area can that treat? Or how far beyond a treatment area can you expect to get some kind of residual effects through a bait application? All right. Shifting over to the field component of the research framework. This is specifically geared towards bait development and improvement. One of the assumptions that practitioners made in the past was that all, quote, fire ant baits would work on little fire ants. And that was clearly wrong for a variety of reasons. Some of those reasons were addressed with the development of the HAL gel bait, but not all of them. It was apparent that some fire ant baits were just completely unattractive to the little fire ants. When we looked closer at those products, we noticed that the unattractive baits all contained insect growth regulators, S-methoprene and pyriproxyfen. This was a big deal because at the time, the only baits with these active ingredients were actually allowed to be used around food crops. And in Hawaii, almost everyone has either fruit trees or a vegetable garden in their yard. So it really limited who would be able to use the effective baits. The other issue was that we were working towards getting a product with S-methoprene, this Tango, this product over here. We were working on getting it registered for public use in the Hawaii Ant Lab gel bait. We had to figure, because of these things, we had to figure out if chemical repellency was a problem with the unattractiveness of the baits, and if there was any way to mask the presence of repellent substances, if repellency is the problem here. So to test this, I conducted a field palatability experiment to test the dose response of little fire ants to the HAL gel bait containing different concentrations of methoprene. Our results showed a very clear decline in recruitment and ant recruitment to the bait as the active ingredient concentration increased. And you can see that here. So as it methoprene increases, the number of ants recruiting also decreases. These cards, this is a very good example of what we were seeing. Once we identified the chemical repellency, we were able to mask the presence of methoprene by including a protein adjuvant into the bait. So the protein adjuvant, it's just another ingredient like torula yeast, peanut butter, beef liver powder, something that was very high protein, but we only added just a little bit. And it was able to mask the presence of it and really boost attractiveness of the bait despite the inclusion of that repellent active ingredient. So that was great. So the final aspect of the field component of my my thesis right now was we decided to do a field demonstration and focusing on trying to eradicate little fire ants in a historically infested site. And so between the work that I was doing on the bait development and work from the past, we were able to get 2 different products registered for use in the HAL gel bait and be able to put forth and use during this demonstration. So developing an ant management plan, we had to do this clearly. Obviously, we had to develop a plan before we actually attempted to do an eradication. You can't just jump in without a plan. That's kind of how things fail. So in order to develop this ant management plan for the field demonstration, it was kind of like baking a mental cake. All of this knowledge that we've gained about the target pest from past and present studies and from unpublished data are basically your ingredients, and then you combine that with the tools that you have on hand. So the HAL gel bait, commercial baits, other potential products, and the knowledge that you gain from assessing the site. For this demonstration, part of my thesis, we developed the little fire ant eradication plant for a historically infested site on the north shore of Kauai. This infestation had seen 2 previous eradication attempts fail, and that was years before. Fortunately, the Department of Agriculture was able to contain it in the meantime, while better methods and strategies were being developed. And that was no small feat on their part. Since we were able to figure out the problems surrounding using insecticidal baits, you know, not being able to stick in the trees, the weathering, the palatability stuff. So we felt that we had the tools available for a successful eradication attempt, but we had to think about the site. You know, no 2 sites are the same, and you have to think about how the ants behave and interact within your specific site in order to really come up with a plan. You can have the tools available, but if you don't use the tools correctly, then it's not going to go anywhere. It's not going to work. So the site that we were working with was complex. And so we had to consider or look at it from a perspective of not like a single site, but kind of break it down into subsites or like sites within a site. The different features of the infested area would be considered a quote subsite, and trying to figure out how we can address each one of these different things individually. The site assessment allowed us to do that and allowed us to theorize not only how the ants are interacting within each of these features, these subsites, but also how the ant baits and the insecticides would likely quote, behave. And that is really important. You don't want to be, you know, applying a granular bait on a steep slope near the coast because of the potential drift into the ocean or waterways. Those are things we had to think about. We couldn't physically get granular baits up into these extremely tall palms. If you can see my cursor, there's a tiny little guy at the bottom of these palm trees. And so these palm trees are at least 50 feet tall. These are huge. So we had to think about not only how the ants are interacting with these systems, but also how we can expect the products to be interacting with the system and how to make this all work. The features identified during the site made us realize that a 2-phased approach was absolutely necessary, and that we'd have to get creative in order to ensure that the entire infestation would be sufficiently treated and monitored, because it's not just about the treatment. We also have to do follow-up monitoring to make sure that it works. The first phase was all of this, you know, it's easily walkable area, it's accessible. This is around the residences, dense landscaping, tall palms. The phase 2 was extremely steep slopes. Excuse me, there were cliff faces, coastal habitat, these rock outcrops and islands. All of this had little fire ants, and we had to be able to think of these things independently. This two-phased, so each phase of the treatment plan consisted of 8 bait applications at 6-week intervals using the HAL gel bait. But it was vital for us to adopt an adaptive management strategy and not be overly rigid in our methodology. Despite the initial site assessment, there were many other obstacles that were identified along the way and during the treatment and monitoring process that required us to make small adjustments here and there in order to address those things. For example, not related to these particular images, but for example, during phase 1, there were residents that were being severely impacted by the ants, and we had to include application of granular baits around the dwellings in order for the residents to get relief during the project. Whenever possible, it might not be ideal. It wasn't ideal to be using these toxic bait, granular bait, while we are doing these growth regulator treatments. It's basically counterintuitive to do that. But if the residents didn't see some kind of relief, they might not have faith in us that the treatments are working, and they can revoke property access anytime. So it can't be overstated that maintaining good relationships and making small adjustments to maintain good relationships with property owners is part of this process and should be considered in your management plan. It can't just be narrow. You can't be narrow-sighted on these kinds of things. The status for this project is basically it took 2 years between the first treatment phase and the last treatment phase, or the last treatment of phase 2. And the site has been in post-treatment monitoring ever since, with since 2016, with 2 to 3 surveys a year until COVID hit, and now it's pretty much shifted to about once a year. The little fire ants have not been detected in the area since 2019. What's interesting is it was easier to get rid of them from the more difficult terrain than it was from the phase 1 area that was easier to navigate. So even though once there has been no detections of the little fire ant for 3 consecutive years, we can start to consider declaring them eradicated from this site. So basically it would be, we're coming up to the 3-year mark this September. If we don't find LFA there, we can consider declaring the site eradicated, but we need to really remember from past experiences that the little fire ant infestations, like historically, they've been brought down to below detectable levels in the past, and for 2 or more years, only for a hot spot to just pop up and be detected, and restarting that proverbial clock. So we can't just say, okay, it's eradicated after September this year. We have to go back and we have to make sure for posterity to keep monitoring, at least for, I would suggest at least a few more years, to be confident that it really is gone. So where does all of this take us? Really, the little fire ants continue to spread throughout Hawaii and the world. There is no end in sight for applied management research. The framework that I've referred to throughout this seminar can engulf an entire career, and there'd still be more work to do. I am looking into testing other active ingredients, such as organic chemicals, since there's no registered organic products available to organic farmers. We're working with product manufacturers to get other products registered for use in the gel bait, because, you know, although it's great to know what works and what doesn't, the knowledge doesn't do the public any good if those tools are inaccessible. We are working with collaborators such as the Maui Invasive Species Committee to develop other application and delivery systems like aerial application for sites that are very large and or inaccessible. And I'd like to be able to work more on better detection methods that could improve the accuracy of our monitoring efforts and thus shortening the time frame for eradication and control projects. One of the biggest takeaways from this thesis seminar is really the concept of adaptive management for little fire ants. There is no silver bullet or any single treatment method that is best for all sites and all situations. Just like no two sites are the same, no two countries or states are the same. Little fire ants are known to behave, to have this behavioral plasticity that, depending on the climate and the ecosystem, they may not behave the same way. And this has to be considered when developing a management plan for this species. Additionally, not all countries have the same access to resources or laws surrounding pesticide use. Solutions have to be, whatever solutions that come up, they have to be accessible and they have to be viable. So the more options that we can explore and validate or invalidate, the better off the world is going to be in the battle against little fire ants. And so this kind of research is going to be ongoing for as long as the foreseeable future. And with that, I know I breezed through everything, but I wanted to have time for some discussion or questions at the end. So thank you very much.
Okay, thanks very much, Michelle. You've done a mountain of work there. There's no doubt about that. We've probably got 5 or 10 minutes for some questions. I'll try and work out from the screen who's got a question. If you can put your hand up to indicate that. All right, I can't see any questions out there. If anyone's got one, speak up now. Otherwise, I'll ask you, Michelle, 2 and a bit years of no LFAs on your eradication attempt. How long do you think you need to monitor for to be confident that it's been successful?
Historically, when it comes to eradications, there's been this 2-year mark that people like to say, well, if you haven't detected them for 2 years, you're gold. You can go ahead and declare eradication and move forward. This has not been sufficient for little fire ants. And we see this time and time again. We've kind of extended that to a 3-year mark, but this is really going to be kind of the first time that 3-year rule has really been pressed. So I would say we could consider declaring eradication at that 3-year mark if we don't detect little fire ants during this during the next survey in September, October. But even if we don't find it, I think it's very important that we continue to go back at least once a year to the site to do full monitoring within the original infested area and beyond, just to make sure that that 3-year mark is sufficient, because it's just we kind of pull that out of thin air a little bit. All we did is really extend the what has been deemed to be insufficient.
And so is there resources dedicated for adequate monitoring for this? One of the great flaws in adaptive management is that monitoring tends to fall off the back of the truck.
Right.
What's the case in Hawaii?
Ooh. It's right now we have funds, and we have the good thing about that particular project is it's a multi-agency project. So it was we at the Hawaii Ant Lab, or I led that project, but we worked in collaboration with the Hawaii Department of Agriculture that's stationed on Kauai, and the Kauai Invasive Species Committee, another group, and they're the ones doing right now, since we haven't been able to get over there due to COVID. They're the ones that have been doing the bulk of the monitoring. So fortunately, we're able to spread that funding over 3 different agencies, but it's not, there's no bottomless pot of funds for anything. And so all good things have to come to an end. And as their target focuses shift to other invasive species, then we will eventually be looking at how to scrape up more funds to continue on that monitoring. And we'll have to justify that. So it is a big concern, and it will be a big concern for all eradication projects.
So there's a question on the chat here. How do you distinguish between an unsuccessful eradication and a reinfestation if they are non-clonal?
That's an excellent question. Sometimes you can't. It depends on, you have to think of what the site usage is. So where did the hotspot pop up? Is it a place where there's been a lot of activity? Maybe there's been items brought into that area, or is it pretty much being left alone? So if it's in an area that's really been left alone, there's not likely to be anything being brought in, then that's when you would probably lean more towards this is a flare up from a residual colony that was unaffected, that survived those treatments and had been undetected for that long. If it's in an area of high activity, then you can't discount the possibility of it being reintroduced. And that is an issue when you have sites like, for example, sites on the Big Island or Oahu that have numerous infestations. It's possible that people are moving things in and out. And we do see that on Oahu, on the southern area, that we get, I mean, we're always getting flare-ups popping up in these areas that we've treated time and time again, and have been ant-free for a long time. So, you just kind of gauge the site as they come.
Okay, I can't see any more hands up on the screen. Is there one final question for Michelle before she has to bolt? All right, if not, can I ask you to join me in thanking Michelle for a great talk, and we hope to see the final thesis soon.
I hope you see the final thesis soon too.
Okay, thank you everyone, and thank you, Michelle.